Hi guys, Dr. Orlando here with a video on meiosis. I'm going to go through the purpose and the steps of meiosis. Okay, so first, meiosis, which is nuclear division to produce eggs or sperm, gametes, kind of basically looks like this. So you take a cell, a 2N cell, remember 2N means it's diploid, so it has two sets of the chromosomes in its nucleus. So this would be a diploid germ cell. Okay, and then you take another diploid germ cell from a different critter. I guess it could be the same critter as well. But either way, one is going to produce one of these germ cells will be responsible for producing haploid cells that are sperm. Okay, and one of these will be responsible for producing haploid cells that are egg. Okay, or ovas. All right, and those will come together. The egg and sperm will come together to make the zygote, which is a 2N cell. 2N diploid zygote. So this will become the baby, okay, or the seed. So that's meiosis in the big picture, the purpose of meiosis. So the goal is to make sperm and egg from two different parent cells, and then to come together to make a zygote. So we're going to look at this process here, going from a diploid cell to a cell that is haploid. Let's put that word in there. Haploid has half the number of chromosomes. Okay, we want to do that. Of course, we have to have the chromosome number, so that when the egg and sperm come together, we end up with a diploid cell again, and then therefore the offspring looks just like, chromosomally, same number of chromosomes as the parent cell. Okay, so we're going to take a germ cell. We're going to actually follow the production of sperm. Okay, so what we're going to do is we're going to take a germ cell, and the germ cell is in the testes. It is a diploid cell, so it has the 2N number of chromosomes. If this were a human, that would mean there are 46 chromosomes in this cell. So let's draw the nucleus on this cell. We are not going to make this a human cell. It's not going to have 46 chromosomes because we can't draw, well, I could, but it would take a long time for me to draw out all of those 46 chromosomes going through meiosis. So instead, we're going to make this little critter is going to have 4 chromosomes in its diploid cells. So we're going to draw them out, and we're going to have a large red one, a large blue one, a small blue, and a small red. So we'll follow these guys, these chromosomes, through meiosis and continue using that color pattern. Okay, so this is our cell. This is the one we're working with. This cell, 2N equals 4. Okay, so that's its chromosome number in the diploid state. So this cell is going to get a signal to do one of two things. If it gets a signal to replicate itself, it might go through mitosis. So if it goes through mitosis, it will make a copy of itself. It will make a clone, right? It'll split, really. So there'll be 2 of them. Why would it do that? Well, we need a bank of germ cells so that we can always be producing gametes. This cell could also get a signal that would tell it to go through meiosis. And in meiosis, this cell is going to split into 4 haploid cells. I'm going to draw him as little sperm here. Okay. And these will each have the N number of chromosomes, which when this is all said and done will be 2. Okay. And if that germ cell goes through meiosis, that particular germ cell is done. It can't then go through mitosis later on. Okay, so these germ cells will get the signal to do either one, mitosis or meiosis. Okay, if it does get the signal to go through meiosis, then it'll go through each of the steps, which is what we will do next. Okay, so if a cell is to go through meiosis, or really even if it's supposed to go through mitosis, it has to replicate its DNA. So its DNA initially will look like this. It will have a red long, a blue long, red short, blue short chromosome. Notice that they are in the single chromosome state. So this would be interphase when the cell is just hanging out waiting for its instructions, whether it's going to go through mitosis or meiosis. If it gets that signal to do either one, it's going to have to replicate its DNA. Okay, so it's going to make those sister chromatids. And when it does, of course, we're going to go through, it would be in G1 at that point. It gets the signal to go through replication, that's the S phase. From there, it's going to go into G2 and make sure that it's got its centrioles and its spindle ready to go. And now it knows it's time to go into meiosis. And the cell would look like this. Okay? All right, so now our cell has had its DNA replicated. We can go ahead and put some centrioles in here as well. It's got 2 sets of centrioles, because it's going to go ahead and split. Okay, remember the centrioles are that kind of barrel-shaped structure that we drew on the board in class, and in fact it's like a protein, and it's there to be the base and the motor for the spindle apparatus. Okay, so now on to meiosis. Now meiosis in fact has 2 phases. It has meiosis 1 and meiosis 2. So we're going to start with meiosis 1, and you use the Roman numeral number 1 to denote that. And it still has those phases that we talked about in mitosis, the prophase, metaphase, anaphase, telophase. So we're going to go through each of those, but remember we have to put the Roman numeral 1 after it and say what's happening in each phase. Okay, so we're going to start here in prophase 1. Okay, so in prophase 1 you can see that I've already done some things here. I've drawn it such that it's already really in prophase 1, beginning of prophase 1. So here's what happens. Let's move this up a little bit and make them just a little hair bigger. Okay, so in prophase 1, initially the nucleus is going to begin to break down. Okay, the chromosomes are condensing, and I didn't draw them all loose like chromatin, but in fact before they come to prophase they are kind of loose like chromatin. They have to condense down into that X shape that we see. But you can see that I'm making my nucleus begin to disappear. The spindle, let's go with orange for our spindle, is going to start, it's going to start assembling. Okay, so this would be early prophase 1. Then we can move on to later prophase and see what it looks like. So here we are in prophase, the middle of prophase, and we can see that the homologous chromosomes are beginning to pair up. What does that mean? So if I zoom in on this guy here, you can see my big blue chromosome and my big red chromosome. These guys are homologous chromosomes. That means that they are carrying similar genes. Maybe not exact copies of the genes. These are from two different parents. The blue one, we'll say, was from this person's father, and the red one from this person's mother, or critter's mother. And so they're carrying similar genes. They're carrying the genes for eye color, for hair color, for certain proteins. Now, maybe there's slight variation in those genes, blue eyes, blonde hair, brown hair, but they're still carrying similar information. So we say they're homologous. They're about the same size. The centromeres are in about the same position. Okay, so the homologous chromosomes are going to pair up, and they are going to do something called crossing over, which you can see here. So in crossing over, a little bit of a part of the one homologous chromosome lays over top of the part of another homologous chromosome, and they form what's known as a chiasmata. So I zoom in even. It won't let me zoom anymore. So let's get another color here. We'll go with purple. So that little area right there, that's the chiasmata. Where they are crossing over and beginning to exchange chunks of their DNA. Okay, let's zoom this down now. I'm going to undo those just so it keeps it neat. Okay. And I'll draw it out. All right, so here's our crossing over occurring between our homologous chromosomes. Good. So what's going to happen in the next picture is we'll see that a little bit of the red chromosome will be on the blue chromosome and a little bit of blue chromosome on the red chromosome. So exchange chunks of their DNA during crossing over. Let's make sure and write crossing over here, because this is a very important part. Pairing up and crossing over. Okay. All right, so give me a second and I'll get this one drawn. All right, so here we are, still in prophase, and this would be what I would call late prophase. So let's label that late prophase 1 of meiosis 1. And we can see that the spindle centrioles have made it to the poles, and that the fibers are here and attached, and we can see that the chromosomes are beginning to attach to the spindle fibers. Also notice that I made my large red chromosome and large blue chromosome. I drew them to show that they were crossing over. Let's zoom in just so you can be sure to understand that. Notice that the red chromosome has a little bit of blue DNA on it, and that the blue chromosome has a little bit of red on it. So exchanging same areas of the chromosome. So we won't be missing any genes at all by crossing over. It just adds adds genetic variability to the process of sexual reproduction. So notice how I have them not lined up yet. They're just kind of being pulled into place, kind of a tug of war to try to line them up on the equator. So let's look at what happens next. So after prophase comes metaphase. So here we are at metaphase 1 of meiosis 1, and you can see that the homologous chromosomes have lined up across from each other along the equator of the cell, okay? Or the metaphase plate as it is called. Now notice they're in a double file line, and they are across from their homologue. That's different than mitosis, where we saw them in a single file line, right? So notice the order. I don't have all of the red on one side or all of the blue on the other side. It's a random assortment. So these two guys will pick a side. We'll just say top or bottom. So top or bottom. Doesn't really matter which one they choose, but they have to be across from each other. These guys will pick a side. Top or bottom, doesn't matter. As long as they are across from each other on the equator, that's the only rule that we have to have. Now think about that. What that means for these guys, it means a little bit of variability, right? If we put all the red on one side, any of the chromosomes that go to this side of the cell are going to be more like mom. And any of the chromosomes that go to this side, if we made it all blue on that side, would be more like dad, right? So by shuffling them up a little bit, we get more of a mix. Now imagine 23 pairs in a human lined up, and every pair of homologous chromosomes can do this top or bottom thing. So huge amount of variability is gained from this this random assortment, and it actually follows a law, so to speak, and it's called the law of independent assortment. So these guys right here being sorted is independent of how these guys are sorted. Okay? And it adds genetic variability. So from here we can go on to the next phase of meiosis 1, and that would be anaphase 1. Slide it over. So here we are in anaphase 1. Anaphase 1 of meiosis 1. And you can see that the homologous chromosomes have been separated from their partner, from their homologue, and they are being pulled towards the poles. Now I drew mine flat, kind of the X's are flat. Sometimes you'll see artists draw them with their legs folded back like that. They're being pulled through the cytoplasm, and that's probably a little more accurate. I'm just not that good at drawing to do that, but it really is being pulled. Remember, those spindles are shortening because of our Pac-Man action. So we've got proteins down here at the bottom that are acting like Pac-Man, and they are shortening. And at the top here, at each of the poles, there are these proteins that are chomping on those fibers and shortening them, Pac-Man style. Waka, waka, waka. Okay? All right. And so they shorten and get pulled to the poles. Very good. Next step is going to be telophase 1. So in telophase 1 of meiosis 1, the nucleus is going to reform, zoom in a little bit, reform around the newly arrived chromosomes. They arrived at the poles. Nucleus reforms around them. The cell is going to begin cytokinesis. So we're going to get that cleavage furrow right there, kind of denting in, and then these purse string proteins are going to start pinching off between the two cells, and eventually we will have two daughter cells completely separate from each other. So when telophase 1 completes and cytokinesis occurs, here we are. We've got two daughter cells. Count the number of chromosomes in each. They each have two chromosomes, so they have the haploid number or the N number, and that equals 2. So we've already halved the chromosome number. But look at the chromosomes. They are still in a duplicated state. So they are not ready to go and be joined up with other chromosomes to make a zygote. We still have to split up those sister chromatids to make it the right form to be an egg or sperm. Okay. So we're going to go on to meiosis 2. Just to mention, notice I didn't draw an interphase between meiosis 1 and meiosis 2. You'll see some textbooks call it interkinesis, the time between the two nuclear divisions. There may be a pause during that time. The cells are preparing, getting their spindle apparatuses and centrioles ready for meiosis II. But we do not, the biggest thing to remember, we do not replicate our DNA in between meiosis I and meiosis II. So we start with a cell that has the n number of chromosomes. So it looks like this. These guys have n chromosomes. It equals 2. They are haploid cells. Okay. The chromosomes are in the duplicated state, though. They have that X shape that we drew because they are still have their sister chromatids hooked together. And the goal of meiosis II is to split up those sister chromatids into separate gametes when it's all said and done. Now, meiosis II, remember, is going to look just like mitosis. So let's go ahead and start. So let's put this cell into prophase. And I'm going to do each of them. This is prophase 2. I'm going to do each of them so you can see the result at the end. So during prophase 2, let me zoom in a bit here, the second set of centrioles will be moved to opposite poles. Do that in both of them. Go that way. This one's going to go that way. Okay. The nucleus is going to begin to break down. Eventually it will be completely broken down. The spindle apparatus is going to start attaching to the chromosomes and begin moving them around. So that once those are attached to the chromosomes, let's draw that out. Once those spindles fibers attached to the chromosomes, they're going to begin playing that tug of war, trying to get those chromosomes to line up on the equator, okay, or on the metaphase plate. Okay, so here we're at the end of prophase 2. We're ready to go into metaphase, so let's go. Notice I did make my cell a little bit bigger. I'm just trying to give myself a little more room to work. There really wasn't much cell lengthening at that point. Okay, so let's go to metaphase. So here we are at metaphase 2. Zoom in a bit here. Centered up. So you can see that the sister chromatids are lined up along the equator, single file line. My spindle apparatus is attached to the sister chromatids, and it's going to get ready to pull. Remember, meta means middle, so think of they're all on the middle line at this point. And again, the single file row, just like mitosis. Meiosis II lines up the sister chromatids along the metaphase plate. So here we are at anaphase 2 of meiosis II, and you can see that the sister chromatids have been pulled apart, and now they are being pulled toward the poles. And I kind of draw them being folded back, like I said in the previous when I talk about meiosis I, they kind of fold back as they're being pulled through that thick cytoplasm. Remember, the Pac-Man action is what is shortening those spindle fibers by these proteins, shortening it and pulling those chromosomes toward the poles. So here we are at telophase 2 of meiosis II, and you can see that the chromosomes have been pulled to the poles, and nucleus has reformed, nuclear membrane has reformed around all the newly arrived chromosomes, and you can see that the cleavage furrow is beginning to happen here, the indentation marking where the cells are going to be split in cytokinesis. You have these purse string proteins that would cinch down and pinch off the plasma membrane between the two cells, creating 4 unique daughter cells. So let's look at that, what the result of cytokinesis here is at the end. So here we are, cytokinesis occurred, and we've got 4 unique daughter cells. I'm going to write this information over here on the side because I'm running out of room. 4 daughter cells. They are all unique. Look at their DNA. Over here we got a large red, small blue. Here we've got a large red with a little bit of blue and a small blue. Here we've got a large blue, small red. And here we've got a large blue with a little bit of red and a small red. So they are all unique. They are all different than each other. Okay. How many chromosomes are in each? They have 2 chromosomes each, so they are half of the chromosome number, so n equals 2. Perfect. They're still haploid, as they should be, and they can go on. If these were sperm, these would go on and mature, and the cytoplasm would shrink down, it would get its tail, it would go through a process to where it would eventually look like a sperm and then eventually be released. Okay, so that is meiosis 1 and 2. If you have any questions, please feel free to contact me. Have a good one.